Nucleic acids. Nucleic acid has two forms. One is the DNA, the other one is the RNA. Structure of DNA is a double strand structure, and then two strands of DNA is facing to each other through so-called Watson and Crick base pairing. Before the DNA structure is fully discovered, Erwin Shagaf has analyzed the DNA structure, and he already knew that DNA is composed of four bases A, G, C, T, and when he analyzed, he found some interesting relationship. Amount of A and T is almost equivalent, and G and C is also very similar to each other. This relationship has been found in other studies, so over and over. So that's why he got some clues. Maybe the DNA has some kind of special symmetry or relationships inside, but he couldn't figure out exactly what that means. Another key information, Rosalind Franklin got X-ray diffraction pattern of DNA molecules. This can give a very important clue about the structure of DNAs. Finally, these two guys, Watson and Crick, they figured out this molecule has a double strand and especially these helical structures of double helical structure of DNA. And this is the first historic paper published in Nature in 1953. So currently we know that DNA molecule has deoxyribose in the center and one side the base and the other side is a phosphate group. Now here is the structure of RNA. RNA usually forms a single strand structure and it does not necessarily form double strand all the time. Now if you look at the DNA and RNA in molecular structure, RNA's sugar is ribose. This is a typical 5-carbon sugar, C5H10O5 the sugar. And deoxyribose is the component of DNA. And two position hydroxyl is the removed. That's why this is called as a deoxyribose. Because of this simple difference, DNA molecule is much more stable than RNA, and DNA tend to form double helical structure, and RNA usually single strand, or it forms some local secondary structures. Now, if you look at the bases, base molecule is another term, alkaloids. These molecules are nitrogen-containing molecules, and there are four different bases in DNA, A, G, C, T, and then RNA utilizes U. And these molecules are connected to ribose or deoxyribose through specific other position. And these are the chemical structure of all bases. And AGCT is utilized by DNA, and in RNA, instead of a T, they utilize the U as the base. As you can see here, each basis it forms this kind of Watson and Crick interactions. A and T make this two hydrogen bonding the connection linkage, and G and C make a three hydrogen bonding linkage. That's why GC pairing is stronger than AT pairing. So through these specific interactions, now we can see those one strand of sequences, then we can read the other side of a sequence depending on these relationships. The double helical structure of DNA has this kind of the structure, and each base pairs they have this parallel linkage, Watson and Crick base pairing, and then they form this kind of helical structures. If you look at DNA's the motion movie, now you can see one strand of the minor groove, and then opposite side there is a major groove is flowing down through this the double helical structures.